now we're going to look at the countercurrent multiplier that takes place in the kidneys. So the main concept for this is for the regulation of the concentration and volume of urine, right? So when we look at the concentration in terms of the different salts, different metabolites that are present, and volume when we're talking about whether there's an absence or the presence of certain volumes of water. Now, when we look at the kidneys from before, this is a nephron. And in nephron, there's two specific regions. We look at the outermost region. This is called the cortex. This is where we have, for example, the glomerulus, where filtration begins. That's surrounded by this structure here. What was that called? The Bowman's capsule, which goes to the next structure. Tell me. The proximal, because it's close. And then we go to this loop of Henle, which is part of a different region of the kidney, of the nephron. Again, this other region here, the distal convoluted tubule, because it's far. And then we have the collecting duct. All right, so this is the cortex. This is the outer layer of the medulla, of the kidneys, excuse me. And the next region right here we have is called the medulla. So the medulla is where we have majority of our loop of Henle. So if you remember this little loop here that's taking place, it's called the loop of Henle. And in the medulla, it is very, very salty. The reason why it's very, very salty is because there is a high concentration of these different minerals, specifically these ions. All right, and again, I'm not going to go over what's going on with these different functions. You should have learned that from the previous videos. But here, when we want to look specifically at countercurrent multiplier, this is a gradient that's created based off of adding or subtracting salts versus adding and subtracting water. So if we go back to the basics, when we look at the loop of Henle, all right, so before we look at the proximal convoluted tubule, what usually happens here? You usually have sodium and potassium reabsorption. Again, other metabolites also is taking place, but you notice that majority of these ions go into the extracellular fluid in the interstitial space. So they're all outside. So anything outside of the green, outside of this pathway, is referred to as extracellular fluid. And so when you have a lot of these specific ions that's taking place, you create this osmotic gradient. This leads to this countercurrent multiplier in order to maintain this osmotic concentration. So if you remember here, so for example, you have some sodiums coming in, potassiums coming in. Let's just keep it at that for now. Other ions also take place, but let's just keep it these numbers. When you come to the medulla, again, medulla is known as to be very salty, meaning there's a lot of specific ions that are taking place. Because these ions are present now, this causes the actual concentration in the extracellular fluid to become high. The majority of the minerals that are present here are high. And so what happens? Well, when we look at this first region of the loop of Henle, this was called the what? The descending loop of Henle, because it's going down. And what was its function? Its function was to bring in water into the extracellular fluid. Okay, eventually can go to the blood, body, but we're going to focus here specifically in the extracellular fluid. And so how do we say this was done? This was done passively, meaning that there is no form of energy being used. So if we look at, for example, if I draw these little dots in the medulla indicating some kind of ion, what happens? Well, this water, in order to regulate this osmolarity, comes in and forces this dilution to take place in the extracellular fluid. So that's why water itself comes in without any requirement of any kind of active transport, no kind of ATP, no energy. All right? So this comes in here. Water comes into the extracellular fluid, dilutes or concentrates the urine. What do you think? So at this moment, whenever water comes out, the urine becomes more concentrated. So in the descending loop of Henle, there is a phenomena where there is a increase in concentration of urine. So by that I mean there are higher solutes to solvent ratios. Now if you come to the other side, again, we're still in the medulla. Medulla is known to be very salty. Now you have water present here. So you have water molecules here that are present, so we can draw them here. All right, so this is water. This is how it looks like. Got partial negative charge, partial positive charge, whatever. Now, what did you remember from the ascending, going up? Ascending had specific transporters that were active transporters. So this was responsible for bringing sodium, chlorine, and also potassium into the extracellular fluid. Now, when there is a very dilute material based off of this phenomenon, this causes the ascending loop of Henle to begin actively transferring sodium, chlorine, and potassium into this medium. So we want to add more salt, okay? Because if, for example, there is a passive process where there's water, too much water is being pushed out. Remember, in the medulla, you want to have a very, very high concentration of minerals. When there is a low concentration of minerals, there is certain metabolic processes that activate the ascending loop of Henle's channels, these active transporters, to push sodium, chlorine, and potassium back into the extracellular fluid. All right? So then what happens afterwards? Well, then whenever you come here, now you have a highly concentrated or diluted urine. So what happens to the concentration as we go towards the distal convoluted tubules? So here, coming from the descending, the urine becomes very concentrated. You're losing water. But now whenever you come through the ascending, you're losing salt. So it becomes more dilute. You're losing the actual minerals. So then by the time it reaches the distal convoluted tubule, again, we still have more reabsorption that takes place. So for example, sodium can come back in, but also, if you remember, potassium goes back into the distal convoluted tubule in this case. And based off of that, that was the antiport that exchanges sodium for potassium. All right. What was that hormone that binds to this region? Aldosterone. So aldosterone binds to this region and it regulates this problem. And eventually it goes out. So this countercurrent multiplier is specifically located in the medulla of the kidneys. And in the medulla of the kidneys, you have this change that's taking place, right? So whenever there is excessive minerals being produced in the cortex, which eventually migrates to the medulla, you have this passive transport of water, which eventually leads to an active transport of our salts. Again, this is a constant circle that's keep happening over and over again, and this is why it's called the countercurrent multiplier.